Erythropoiesis is the process by which red blood cells, erythrocytes, are produced in the bone marrow, and it is tightly regulated to maintain oxygen delivery to tissues. And erythropoiesis is a very important phenomena of the red blood cell, because it's the process of making the RBC, which are very important as they carries oxygen and deliver it to the tissues, so that our organ get oxygenated. Like we need uninterrupted oxygen supply to heart and brain, right? So and RBC also has a lifespan of 120 days. That's why after 120 days our RBC will die. So we need to produce new RBC. That is why we required erythropoiesis, which occurs within the bone marrow, specifically red bone marrow. So the first stage of erythropoiesis is the stem cell differentiation. What is it? Is a stem cell differentiation. Okay. So in stem cell differentiation, firstly we have pluripotent uncommitted stem cells. Like these type of stem cells can give rise to many different cell types, many different it can generate many different organs. Like it has the potency, ability to generate into different type of cell types, but it cannot generate the embryonic cell types. That is why they are pluripotent. The stem cells, they which have the ability to generate the embryonic cell stages, they are totipotent. So these are the pluripotent and uncommitted stem cells, because they can give rise to many type of organ, like heart. It can also generate bone marrow. Okay, whatever it can be. Now these pluripotent uncommitted stem cell give rise to hematopoietic stem cells, and they are the multipotent stem cells. They differentiate and they produce the multipotent progenitors. So hematopoietic stem cells in the bone marrow differentiate into multipotent progenitors. And progenitors means the which form the progeny, initiate the generation, okay, generation of RBC. So that is why they are progenitors, multipotent progenitors. These progenitors committed, now they commit that they will only give rise to only one type of cell types, that is RBC. Okay, so these progenitors commit to the myeloid lineage, because it can give rise to multipotent progenitor, can give rise to myeloid and lymphoid. It can give rise to two different cell types, like lymphoid and myeloid. Myeloid can give rise to RBC, WBC and platelet. That is why we are choosing myeloid progenitors, and multipotent progenitors commit to the myeloid lineage, leading to the production of erythrocyte precursors. Okay, so the next step is erythroid progenitor formation. The first cell line of the erythroid progenitors are burst forming unit erythroid, like burst forming unit erythroid. So they are burst forming unit erythroid. This burst forming unit erythroid is named as BFUE. Okay, BFUE, these are the precursor cells. BFUE cells differentiate into colony forming unit erythroid. They differentiate into colony forming unit erythroid, that is CFUE. Okay, and these cells, colony forming unit erythroid cells, under the influence of erythropoietin hormone EPO, okay, produced by the kidneys in response to low level of oxygens. So here some stimulators like erythropoietin hormone, and which is produced from erythropoietin, which is produced by kidney in which condition? Low levels of oxygen. Low levels of oxygen, or when there is the hypoxic condition, that means there is no oxygen, insufficient oxygen, like a person is traveling high altitude, okay, so in that case they are facing hypoxic condition. At that time erythropoietin will release from the kidney and it will stimulate colony forming unit erythroid cells so that they can generate RBC. Okay. So next are the erythroblast stages. CFUE cells further differentiate into erythroblast. So before erythroblast we get proerythroblast, and this proerythroblast is the earliest recognizable cell of the erythroid series, okay, and it is seen in the red bone marrow. It is seen in the red bone marrow and red bone marrow of flat bones, like it can be seen or produced within the proximal end of long bones. It will be proximal end of long bones like femur, and for flat bones it will be sternum, ribs, okay, brain cavities, and also pelvis bone. So these are the proerythroblast, and the cell size is large for proerythroblast. There are some characteristics of the proerythroblast cells, that the size is large with a diameter of 15 to 20 micrometer. So the cell size is large, okay. Large cell and the nucleus is also large, okay, nucleus is present. So large cell with a nucleus, because at this stage here occurs the DNA replication, and mitosis is seen in these cells because we need to produce a large number of erythroblasts. So we have to increase the cell number. So cell division is needed. That is why there occurs mitosis. So what are the characteristics is seen in the proerythroblast? That these are the large cell with nucleus is present. These are very important, and here occurs mitosis and DNA replication, because if there is DNA replication, then definitely there will be RNA synthesis. And why we need RNA synthesis? For protein synthesis. And which protein is present within the RBC? That is hemoglobin. So for synthesis of hemoglobin we need RNA synthesis, okay, because RNA with the help of ribosome can synthesize protein for RBC, which is a hemoglobin, okay. Next, but in proerythroblast there is no hemoglobin, so I will not write hemoglobin in this stage, because later you will get confused that maybe in this stage we can see the hemoglobin also. But no, proerythroblast, within proerythroblast there is no hemoglobin present. Okay, hemoglobin is absent in this at this stage, and mitosis is seen in these cells, and there is a nucleus and a deep basophilic cytoplasm, because of the high content of RNA, because there is DNA replication. So there is also DNA replication, and there is also RNA synthesis. That is why blue color cytoplasm, blue color cytoplasm. Maybe these cells are look like this way. They are large, as they have the diameter of 15 to 20 micrometer. Okay, and they are having a big nucleus. They are having a big nucleus, and within this nucleus there is nuclei and nucleoles, and chromatin also present within this nucleus. Okay, and these chromatin are basically the euchromatin, because they are loose chromatin. So within this nucleus is present, and nucleus houses loose chromatin, loose chromatin, that are euchromatin, that are euchromatin. That are euchromatin. Okay, and here also synthesis of, if there is nucleus, definitely they will synthesize some RNA. Okay, because, and ribosome. So here will be ribosome, because nucleus synthesize ribosomes. So there is ribosome, and also nucleus will synthesize RNA. So there is also RNA. So RNA, basically these are mRNA. These are mRNA, messenger RNA, with the help of ribosome. Messenger RNA with the help of messenger RNA with the help of ribosome, they will synthesize protein. But for proerythroblast, there is absence of hemoglobin, and the cytoplasm appear blue color, because they are basophilic. Okay. Next, the basophilic erythroblast. Next is the basophilic erythroblast, and this basophilic erythroblast, here occurs the synthesis of hemoglobin started in this stage. Okay, so cytoplasm stains blue due to RNA content, because there is also RNA present. Okay, so cytoplasm is still basophilic, and nucleus is still large. Okay, and hemoglobin synthesis just begins at this stage. Hemoglobin synthesis at this stage. Okay, and cytoplasm stains blue due to RNA content. Cytoplasm stains blue due to RNA content. Cytoplasm stains blue due to RNA content. Okay. Next stage is the polychromatic erythroblast, and I have written this stage in bicolor, like P is in blue color, O is in red color. Why? Because at this stage hemoglobin synthesis is high. Okay, so increased hemoglobin synthesis, and hemoglobin is red color pigment because of it contains iron, so it's red color. So here occurs the increased hemoglobin synthesis, increased hemoglobin synthesis. So this cells turns little red color. Okay, but its cytoplasm is blue color, so that I have written it's blue and red. We have seen two color in this stage, like blue color cytoplasm and red color hemoglobin, so that there is two chrome. Chrome means color, poly means more than one, so more than one color is present. That is why it is named as polychromatic. Okay, so polychromatic erythroblast, where hemoglobin synthesis increased. Okay, and yeah, here also occurs like cytoplasm little grayish because it's blue and red, so that color changes little grayish. Okay, so increased hemoglobin synthesis and cytoplasm content is blue. Okay, so this cytoplasm is blue color. Now it's orthochromatic or normoblast. And orthochromatic is the last nucleated cell of erythroid series. Okay, because nucleus start disappearing from this cell stage. Okay, so here the cell size is reduced. What are the changes occur in this stage? Firstly, the cell size reduction. So firstly is cell size reduction. Cell size reduce. Next is the cytoplasm is characteristically acidophilic because of the production of hemoglobin. A huge amount of hemoglobin produced, that is why cytoplasm get acidophilic instead of basophilic. Okay, so cytoplasm is acidophilic. Cytoplasm is acidophilic. In this stage, mitosis is absent. Okay, so the cells stop dividing. From this orthochromatic cell stage, there is no further division. So the number is limited from this cell step. Okay, so they stop dividing. Okay, so they stop dividing. Third point, stop dividing, so that mitosis is absent. Mitosis is absent. And also nucleus becomes condensed. Nucleus becomes condensed. Now, what do you mean by nucleus condensation? It means nothing but condensed means nucleus contains chromatin. So here occurs chromatin condensation. Chromatin condensation. So it is known as pycnotic with dark chromatin. With dark chromatin. Pycnotic with dark chromatin, where also in orthochromatic normoblast. It is seen within the orthochromatic normoblast. Okay. And eventually nucleus is ejected out from the cell. And whenever this nucleus ejected out from the cell, within the bone marrow, there is also some macrophage present within the bone marrow. They engulf the nucleus then and then. Whenever they get ejected out from a cell, they are less effective. So less effective thing is get destructed or utilized or removed by the macrophage. Okay. And next is the next cell stage is the reticulocyte. 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 Reticulocyte is the last stage in the formation of erythrocytes. That is why it is also called young red cell, reticulocyte. And why they are named as reticulocyte? Because there is a reticular network of RNA is seen in this cell stage. Reticular formation. Reticular formation of RNA. Reticular means network. A network of RNA is formed within this cell stage. That is why they are named as reticulocyte. That means in this cell stage there is also presence of little amount of RNA, ribosome and mitochondria. Okay, within this reticulocyte, they are still synthesizing hemoglobin. So HB synthesis also occur within this stage, but it is very less amount, and nucleus is absent. The reticulocyte results when the nucleus is extruded from the late normoblast within the marrow. Late normoblast means that orthochromatic normoblast, because it is also named as normoblast. So some scientists named as these cell types like they called this basophilic erythroblast like early erythroblast, polychromatic erythroblast like intermediate erythroblast, and orthochromatic erythroblast like normoblast or late normoblast. Okay. So within the reticulocyte there is absence of nucleus is also absent. Nucleus is absent. Okay. And cytoplasm still contained a small amount of RNA, and cell size become change, and cell size is flat and disc shaped. So that these are the changes that occurs within the reticulocyte. But these reticulocyte are juvenile red cells devoid of nucleus but contains ribosomal RNA, so that they are still able to synthesize hemoglobin. A reticulocyte spends 1 to 2 days in bone marrow and circulates for 1 to 2 days in the peripheral blood before maturing in the spleen. So so it has for maturation, this reticulocyte has to go where? In the spleen. Okay, for maturation. For mature RBC, it has to move towards spleen. Okay. But and to become a biconcave, to achieve its biconcave shape, because we know that red blood cell is biconcave shaped. Okay, so to achieve biconcave shape and to remove the other organelles like ribosome, mitochondria, it has to go the spleen. Okay, so the mature red cell has the mature red cells has lost its nucleus as well as the ribosomes and mitochondria. It thus cannot synthesize hemoglobin and any other protein. The immature cells in various stages of development are found outside the blood sinusoids of the bone marrow. Normally, only mature blood cells are able to enter the circulation. Okay, and some important factors are necessary for erythropoiesis, that is the first in the first stage, that is the erythroid stage, that is the proerythroblast. In this stage we need vitamin B12 because vitamin B12 is a necessary component for the formation of DNA. Okay, so DNA formation we need vitamin B12. Vitamin B12. If there is absence of vitamin B12 or vitamin if a person is getting malnutrition because of vitamin B12 deficiency, or there is any problem in absorption of vitamin B12, that may lead to degeneration of DNA, that may lead to degeneration of RBCs and lesser production of RBC, and that causes anemia. Vitamin B12 deficiency causes megaloblastic anemia because of the impairment of erythropoiesis. Okay, so it causes megaloblastic anemia. Megaloblastic anemia. Megaloblastic anemia. Okay, and the other important factor or regulator of erythropoiesis is um, erythropoietin, which is a very important component. If there is absence of erythropoietin hormone, because within this CFU there is the erythropoietin receptors are present within the CFUE. They are houses EPOR, erythropoietin receptor. So this erythropoietin binds with this EPOR, but if there is deficiency of erythropoietin, it cannot stimulate the colony forming unit erythroid cells, so there is no further generation of proerythroblast. Okay, and we all also know that for hemoglobin, for its red color, we want iron. Okay, Fe2 plus. So if there is a iron deficiency, that may lead to hemoglobin synthesis impairments, and that will lead to the impaired formation of red blood cell. So erythropoiesis also get hampered in deficiency of iron deficiency. Okay, so these are the factors and for erythropoiesis there are some genetic cause, some signaling pathway, JAK2 STAT5 pathway. Okay, and some genes are responsible for synthesis of hemoglobin, and so there are some genetic reason, also some signaling, molecular signaling also involved within erythropoiesis. But the overall idea of erythropoiesis is about like whatever I have discussed in this video, and hope this video is helpful. It will help you to understand erythropoiesis in a brief. So there is a diagram of erythropoiesis, the changes of cells like proerythroblast, This one is the first stage of erythroblast formation. Then see, it has a bigger nucleus, and the cytoplasm is like purple color. Then it turns light slight red cytoplasm because hemoglobin synthesis starts within basophilic erythroblast. Okay, and then there is a polychromatophilic erythroblast where the hemoglobin synthesis is high, and this orthochromatic erythroblast where the mitosis stops, cells stop its division, stop dividing, and nucleus is become condensed so that it will ejected out. See, nucleus is ejected out, and this nucleus, after its ejection, it is removed by macrophages. Macrophage are the cells, they are already wandering within the bone marrow, and they come and eat this nucleus. Okay, so a reticulocyte is without nucleus. Then it also loses is another organelles like ribosome, mitochondria, and ultimately it forms erythrocyte. Okay, hope this video will be helpful for you all, and if you like my video, then please do like, and stay tuned with me, and I will meet with you in the next class.